Hi guys, welcome back to my YouTube channel. I know that I've been missing for maybe about 2 months. So today I'm back with a subtopic that a lot of students don't understand the most, which is Form 4 Chapter 6. I'll be doing mitosis today. Maybe next video I'll be doing meiosis. Personally think that Form 4 Chapter 6 requires a lot of drawing, a lot of understanding instead of just memorizing, because if you memorize, you don't get the concept. So you need to practice, to draw, and to understand how does mitosis and meiosis works. So today we're going to start with mitosis first. Next video, I'm going to continue with meiosis. Hopefully the next video is not another 2 months. Okay, probably next few weeks I'll be coming out with meiosis video. Okay, so let's get started with the class, which is the screen over here. Now we're going to start with mitosis importance first. Where does mitosis happens at? Mitosis happens at the cell called somatic cell. What is somatic cell? Somatic cell equals to body cell. All of our body parts, we have body cell. Besides reproductive cell, which is sperm and ovum. Besides sperm and ovum, the rest of body are known as somatic cell, which is body cell. And then it can also happen in plant cell. Whenever you see 2n, right, what does 2n representing? We have 2n and we have n. So 2n representing diploid number. Diploid number, it means full set of number. For human, okay, we're talking about human, okay. For human wise, right, our diploid number is 46, because as a human, chromosome number should be 46 or 23 pairs. We call it as 46 chromosome. So for haploid number, haploid can only happen in meiosis. You will learn this in meiosis. For human wise, it will be 23. Haploid is n, diploid is 2n. This is a fixed number, okay. For human, is fixed number, which is 46 and 23. But most of the time, we have to count based on the diagram above, the diagram given. You have to count based on the chromosome given. Not every time when you see human is 23 and 46. I have a video teaching you guys how to calculate the number of chromosome, a comparison of mitosis and meiosis. I did the video, I think 1 or 2 years ago. So maybe you can watch the video, or maybe last year, 1 year ago, I think. So you can watch the video after you have learned mitosis and meiosis, maybe not now, okay. After you learn mitosis and meiosis, you can watch the comparison video, okay. Now, this is where mitosis happens at body cell and plant cell. Remember, diploid number is 2n. 2n, it means a full set of chromosome. So this full set of chromosome, it depends on what are the example they are given to you. It is either human, is either the diagram given. We usually count with the diagram given. So imagine 2n equals to 46 and haploid number equals to 23. Okay, maybe next week, next video, I'll teach about the haploid number, which is meiosis. Importance of mitosis. Why do you think humans need to carry out mitosis? Mitosis has a similar function as protein. What is the function of protein? To produce new cell, to generate and repair cell for growth and development. If you notice, the functions are the same. You can see repair lead cell, same as protein, right? Regenerate, repair damaged tissue, same as well, right? To allow growth and development, also same as protein, right? So if you don't remember the function of mitosis, maybe you can write the function of protein over here. Personally, I don't choose these 3 as my answer when I explain the significance of mitosis. What I usually write in mitosis is I will write to produce genetically identical daughter cell. What is genetically identical? Means exactly the same. We call this as genetically identical. For example, when you're clapping your hand, okay? When you're clapping your hand, right, all of your cell die on your hand itself, right? All of the cell die over here, right? So what happens next is you have to produce a new cell. How do you want to produce a new cell? It's by carrying out mitosis. You have to be the same. So let's say, as I mentioned, human chromosome number is 46 in total, right? If you cut your hand, your cell died. Your cell at your hand should produce 46 as well. Your hand has to produce a hand cell. It will never produce a sperm and ovum cell. So if your hand produces sperm and ovum cell, just imagine if you hold hands with some random people, it means that you're going to get pregnant. If there is sperm and ovum, is it possible? Not possible, right? So it must be genetically identical, exactly the same. Whole hands will never get pregnant, guys, okay? Don't think that, no, you're watching some childhood or cartoon, okay? Whole hand can get pregnant, no, okay? Because whenever we get injured, it should produce the same cell, same as the surrounding cell. Hands is hands, leg is leg, okay? Any part of body will be the same as well, besides sperm and ovum, okay? Next, what I usually write is to maintain number of chromosome in daughter cell. Maintain number of chromosome, what does it mean? As I mentioned, if my hand is 46, then I should produce 46. I will never produce 47, 48, 49. It will always be the same. We call this as genetically identical cell to maintain number of chromosome in daughter cell. I will teach you what is daughter cell, what is chromosome later, to calculate the number of chromosome later, okay? So these are the importance. If you want to write the replace dead cell, repair, regenerate to allow growth and development. It's totally fine, guys. Okay, there are many more significance of mitosis. I'm just giving you a few over here, but the one that I usually write is to produce genetically identical cell and to maintain number of chromosome in daughter cell. This is what I usually, the one I highlighted lah. And these are the answers that your teacher should give it to you as well, which is to produce genetic identical cell, to maintain number of chromosome. Okay, now we have to go through the next part, which is mitosis. Every time when you learn mitosis, I don't know how your school teacher teach you, but the most productive way and the most effective way to learn mitosis is always starts from drawing. If you don't know how to draw, literally you never understand what does the mitosis and meiosis are talking about. So you have to draw when you learn about mitosis and meiosis. No matter how, you still need to draw out, okay? So you can just prepare a page or something like that. Okay, you can draw something like that. Maybe you can pause the video to draw something like that first before we continue. Okay, so for those who have drawn already, then we're going to continue over here. Mitosis, we have 4 phases. The 4 phases, we call this as PMAT, P-M-A-T. What does PMAT mean? P stands for prophase. Let me write over here. This is prophase. Yeah. Okay. Prophase, P, okay. M stands for metaphase. A stands for anaphase. T stands for telophase. So we call this as PMAT. P-M-A-T. The sequence you need to remember. P-M-A-T. P-M-A-T. We call this as PMAT. Okay, let me teach you a very easy way as a conclusion. How do you want to remember them? Okay, remember P is a preparation phase. What does a preparation phase mean? Preparation, everything started to get ready. We call this as preparation phase. And then M, remember as middle. They will be at the middle, okay? Metaphase, middle, okay? Anaphase, you can remember as Kssm. They are separated, okay? Kssm, they are separated. Okay, and then T is opposite. Then prophase. They are opposite than prophase. So this is PMAT. As a general idea, you can remember like that. No matter whether it is mitosis, meiosis 1 or meiosis 2, this is a general idea of how we remember PMAT. Prophase is a preparation phase. Everything started to get ready. Metaphase, they'll be in the middle. Anaphase, they are separated, which is Kssm, okay? And then telophase is opposite than prophase. Okay, so these are the things that we can remember as a general idea. Now we're going to start drawing. Everything we have to start from prophase first. My prefer is when I draw, I will always start from the centrioles, okay? Centrioles at prophase, they will start to move to opposite pole because when we draw, right, we have to show that they are starting to move to opposite pole. So how are we supposed to draw? I personally like to draw either at 9 or 12 o'clock. 3-6 also can. If you want to draw 3-6, 6-9, 9-12 also can. I mean, it's your choice. As you show that they are trying to move to opposite pole, then okay. If you want to draw nearer to each other, like that, also can, no problem, as long as you are trying to prove that they are moving towards each other. So we're going to label this thing as centrioles. Okay, what is the function of centrioles? You learn this in Form 4 Chapter 2, is to form spindle fiber. So to show that we are starting to form spindle fiber, I'm going to draw half. Okay, starting to form spindle fiber. So this is spindle fiber. Okay, every time you draw, you have to label. Okay, next we have to draw a nuclear membrane. At prophase, nuclear membranes start to disintegrate. Means they are breaking down. If they are breaking down, I have to show that they are breaking down, right? How am I supposed to draw? I draw a dotted circle. Because dotted circle, why I don't draw a complete circle? Because they are starting to break down. If you are trying to break down, means that they are trying to break, right? If you draw a complete circle, you're not going to show them they are trying to break. So this is a nuclear membrane. Nuclear membrane, okay, nuclear membrane. Okay, after you draw nuclear membrane, you have to draw a dot. If you're using a pencil, you can draw slightly not so obvious because this, we call this as nucleolus. Nucleolus at prophase, they will start to disappear. So you have to draw maybe lighter if you're using a pencil, but if you're using a pen, it's totally fine, okay? By drawing all these things, actually it's not the main thing, but you have to draw the main thing, which is chromosome right now. Chromosome, you can draw all same size, you can draw all same color, you can draw all different color. It's your choice because this is mitosis, right? We don't really have a standard of how do we have to draw a chromosome in mitosis because they will never exchange information, they will never have any crossing over, so it's your choice. I will draw all same color and maybe different size, okay? Same color, different size. Same color means that either you shade everything or either you leave it blank. I'm going to leave it blank because it's easier. Okay, because if you shade it, it's going to take a lot of time. So I'm going to draw two big ones. Any place that you want to draw also can, and two small ones. Okay, two big one and two small one. Okay, as long as you want to shape them also can. Don't need to be so straight also can. You can be like that. No, you can be like that. And also can, totally fine, because they are still in prophase. So we have to label what join them together. The one that join chromosome together is the centromere. There's a dot over here actually. There is a connection point. This connection point, we call this as centromere. Centromere is to join sister chromatid together, is to join, is a place where spindle fiber will start to attach at. It's called a centromere. Okay, it's the point, it's a dot there, is to join the sister chromatid together. The left and right side should be the same. We call this as sister chromatid lah. Next, this entire thing is called chromosome. Sometimes you can write as chromosome, you can write sister chromatid. Sister chromatid is the one, the left side is the same and your right side is called a sister chromatid. Okay, or we call it as chromosome easier, it's called chromosome. So a butterfly right now is a chromosome, guys. Okay, this butterfly, I usually call it butterfly, it's easier for you to recognize. Okay, the butterfly here is called chromosome. Whenever we draw, we have to know how to form sentence. Literally, just now the one that I mentioned, you just put it into sentences. Number one, we're going to start from the centriole first, because just now we mentioned about centrioles. Maybe I use different color. Okay, maybe I use blue color over here lah. Okay, so you're going to say, there's a centriole. Okay, you're going to start from chromatid first, sorry. Chromatid is the chromosome. Okay, because we have a sequence. Chromatid, it forms into chromatid, TID, it forms into a sister chromatid. Chromatid is a fine thread. This chromatid, right, is so fine that we can't even see under the microscope. They are not visible under microscope, and they will form into a chromatid. They will form into a sister chromatid. A sister chromatid, also known as the chromosome lah. Okay, sister chromatid must be the one on the left side, same the one on the right side. This part here should be the same as this one here, called sister chromatid. So because at prophase, they will start to become more visible. In order to become more visible, the chromatid will condense. What is the meaning of condense? Condense means that they join together. Okay, they are shorter and thicker. Just imagine if the person is short and fat. Okay, maybe it's not good to use this example, but you can imagine that if the person is short and fat and the person is long and thin, like a paper like that, which one is easier to recognize? Of course, it's the one that shorter and fatter, right? It's easier to recognize because they are more obvious. The thin like a paper, how to recognize them from far away, right? If you fat, more meat, maybe easier for people to recognize from far away. Same as this one. They must be shorter and thicker so that they are more visible under the light microscope. Okay, so we're going to write in the sentences. We're going to write chromatid. Chromatid, condensed. If you are able to remember the word condensed, you can just write condensed. But if you don't remember the word of condensed, you can write they become shorter and thicker. Okay, shorter and thicker. And form a chromosome. They are visible under light microscope. Next, sister chromatid join at centromere. Centromere is the dot that is called a centromere. The one that labeled that is called a centromere. Next, nucleus disappear. So that's why when we draw, we have to draw slightly lighter in color because to show that they are disappearing, right? And then centriole starts to move to opposite pole, spindle fiber. Spindle fiber starts to form. And the last one is the nucleus. Nucleus. Nucleus, I wrote already. Nuclear membrane, sorry. Nuclear membrane. Disintegrate. Okay, so many sentences here, right? Which sentence is more important? We usually look at the chromosomal behavior. What is the chromosomal behavior? Is the one that related to chromosome. For example, at prophase, chromosomal behavior will be the first one and the second one. Usually, we're going to write these two for prophase. If you want to write sister chromatid join and centromere, no problem as well. But the rest of the sentence, we don't really write at prophase unless it's an AC question. They ask you to explain prophase. Then you're going to write everything lah. I mean, unless it's an AC question. If it's not, then we can just ignore them. Okay, so next we have metaphase. Just like I mentioned, right? Metaphase means what? Metaphase means they are at the middle, right? Called metaphase, right? So right now we have to draw at the middle. Just now we say, right? The centromere will move to opposite pole. So we have to draw centrioles. We have to say centromere. Centrioles move to opposite pole already. And then we have to draw the chromosome at metaphase. Just now we draw too big, too small, right? Over here, we still have to draw too big, too small. The sequence, if you want to write big, small also can. Small, small, big, big also can. Big, big, small also can. So it's your choice. Okay, I'm just going to draw big, small. Big one, and then the small one, and then the big one, and then the small one. Okay, so big, small. And then we have to draw the spindle fiber. Spindle fiber are from the centriole and they will join at the centromere. Usually I'll just draw like that because it's easier for me to aim the centromere. But if you want to draw from the other way around also can, means if you want to draw from here to here also can, but it's even harder to join at the centromere. It's your choice anyway. Okay, usually for metaphase onwards, we don't have to label anymore because we have label for prophase, right? If you know how to label prophase, then you should know how to label metaphase because it's the same. We have a new thing here that we can label, which is the middle part. This is an invisible line. We don't have to draw it in exam, but it's an invisible line. This invisible line, we call this as equatorial plane, or we call this as metaphase plate or equator. We have three names, okay? But if you follow your textbook, it should be equatorial plane. But we have equator and metaphase plate as well. I mean, it depends on which one your teacher teach you. Equatorial plane, metaphase plate, three are acceptable. So they will arrange themselves at the metaphase plate and spindle fiber attached to the centromere of chromosome. These are the importance of metaphase. So you have to form sentence right now. Okay, whatever you draw, you need to understand why are you drawing this. If you understand you are drawing this, literally you can form a sentence. If you don't know what you are drawing right, then it's very hard for you to understand the mitosis already. Okay, so over here, number one, centriole are at opposite pole. Chromosome align at equatorial plane, equator, or metaphase plate. Spindle fiber are fully formed and attached to centromere of chromosome. Okay, so this is the answer. If you can't remember the word align in an exam, what you can write is you can write line up. Line up also can. Don't need to write align, it's totally fine. Okay, so same thing, we have to find chromosomal behavior. What I see chromosomal behavior? Something to do with chromosome, we have to call it as chromosomal behavior. This one and this one is chromosomal behavior. So if they ask you to explain, or based on the diagram above, explain the chromosomal behavior and the diagram above is metaphase, these are the two sentences that you have to write. Okay, so this is metaphase. Next, anaphase. Anaphase means what? Async, right? They are separated. Okay, they are separated. It's called async. Just imagine right now, this part here is being pulled to the left side. This part here is being pulled to the right side. Or maybe you just imagine you yourself. Your left hand and your left leg is being pulled from your girlfriend number one, okay? And your right hand and your right leg is pulled by your girlfriend number two. So what happened to you? You're gonna break into half because who else you to date two girls, right? That's why so you're gonna break into half, which is anaphase. Now we have to draw that they are breaking into half. Just now we draw big, small, right? Okay, big, small. We have to also draw big, small over here. But since they are being separated from a butterfly, what they will be formed right now? They will form into a fly. Okay, you know what's a fly, right? Housefly. Housefly, something like that. Housefly. Okay, they will form into a fly right now because they are being separated. So same thing, we're gonna start from the centriole first. One on the left, one on the right. I'm not gonna draw the spindle fiber first. I'm gonna draw the sister chromatid first, okay? The sister chromatid will be separated and the centromere will be divided into half. Okay, the centriole on left and right side both also have. So basically, this is the big one. This is the small one. This is the big one. This is the small one. Big, small, big, small. Okay, so you have to follow the sequence that you draw at metaphase. Big, small, big, small, big, small, big, small. Okay, and then we have to draw the lines. Okay, these are the lines over here, the spindle fiber. So remember, what you draw, you have to understand what you are drawing. Okay, you just don't simply draw what you are drawing. You have to understand. Okay, let's form sentence right now. We don't have to label over here. We have to form sentence. Now, when I start from anaphase, right, you have to say the centromere divide into half. These are the centromere, right? This is the centromere, they are divided into half. So we're going to say that centromere divides into, not half, divides into two parts. Or one on the left, one on the right, divides into two parts. Did I say half? Yeah, I think I said half just now. Okay, the sister chromatid separated and pulled to opposite pole by contraction of spindle fiber. Okay, so this is the answer that we have to write. There's one more sentence here, which anaphase is completed when sister chromatid arrive to opposite pole. Same thing, we have to find what? We have to find chromosomal behavior, right? Which one is chromosomal behavior? This sentence here is a chromosomal behavior. Sister chromatid separated and pulled to opposite pole by the contraction of spindle fiber. So without spindle fiber, the sister chromatid cannot pull to opposite pole. So what happens when the spindle fiber are not fully formed? They will be abnormal number of chromosome later on. Means maybe the person will have Down syndrome or some other disease lah. Because the some other disease, you only learn it in Form 5. So Down syndrome will be the best example that you learn it in Form 4, okay? If they didn't separate, it means one like that, right? If they are this one don't have anything, right? Then the number of chromosome will be different already. Okay, next, telophase. So telophase, they're opposite than prophase, right? So since they're opposite than prophase, whatever we draw are similar to prophase, but when we form sentence, it's opposite. So first, same thing, we have to draw from the centriole first, one on the left, one on the right. Okay, you can see they are starting to form into two cell, right? Like a butt shape, right? And then we have the spindle fiber. We have the nuclear membrane. This is the nuclear membrane. Same dotted, okay, because just now spindle fiber starts to form. Right now, spindle fiber starts to disintegrate. Why do we have the dotted circle? Because just now the nuclear membrane starts to disintegrate. Right now, nuclear membrane reform. And then we have nucleolus. Just now, nucleolus disappear, right? Right now, they will reappear again. So the two dots you can draw randomly at any places if you want to. Any up, down also can lah. But the main thing is not drawing this thing, same thing as I mentioned. The main thing is how do we want to draw our daughter chromosome? Okay, imagine this entire thing. This thing move to the left. This thing move to the right. How are we supposed to draw this? Remember, it's big, small, right? So in one cell, we have too big and too small. How do you want to draw these too big and too small? It's up to you. As long as you can show that you have too big and too small. So this one, small, big, also cannot. Your choice. As long as you can show that too big, too small, then okay already. Then we have to form sentence. When the chromatid at the opposite pole, they are now known as the daughter chromosome. What is a daughter chromosome? Okay, later I explain to you after I form all of the sentence over here. It says each pole contains a complete set of identical chromosome uncoil and lengthen to become thin and chromatid threads. Just now it says the chromatid condense, right? Condense means they become shorter and thicker. Right now, opposite meaning uncoil means they will form into a thin and long again. Instead of short and thick, it becomes long and thin again. Spindle fiber, just now it was forming, it will disintegrate. Disintegrate means they will break down lah. And centrioles replicate. What does replicate means? It means every single cell, one cell, we need to have two centrioles. One cell, we need to have two centrioles. So later when they form into two cell, like literally two cell, right? So we have two centrioles again. It's called replication. It's a copy. It's a photosetting lah. I can say it's a photosetting. It's called replicate. Nuclear membrane just now was disintegrating. So right now they will reform around the chromosome. Nucleus just now disappear. Right now they will reappear. The telophase stage is followed by cytokinesis. But today I will not continue with cytokinesis because it's another subtopic. So this is how we draw a mitosis. So again, what is a daughter cell? What is a daughter chromosome? Daughter cell, it means one cell, let me write over here. One cell, they will form into two cells. This is parent cell. This is daughter cell. Okay, we call this as daughter cell. So 1 into 2, this is normal mitosis. 1 into 2, 2 to 4, 4 to 8, we call it as daughter cell. What is a daughter chromosome? Daughter chromosome means that the chromosome inside a cell. So let's say, for example, the parent cell start with 4 chromosomes. If this is mitosis, right, the daughter cell should produce genetically identical cell. What does genetically identical means? Exactly the same, right? We call this as genetically identical. So the daughter cell will have 4 and 4 as well. Not 2, 2, it's supposed to be 4 and 4 because it's genetically identical. So the number 4 or 4, 5, 6, 7, 8, 9, 10, those we call this as daughter chromosome. The one in red color word is called daughter chromosome. Or another example. So let's say the parent cell, we have 8 chromosomes. 8 chromosomes. So the daughter cell have how many chromosomes? Also 8, 8. It's not 4, 4, it's 8. It's not 16, 16 also, okay, it's 8, 8. It will always be the same as your parent cell. Okay, so this is what we call as mitosis. Okay, in conclusion, what do we have to know from this subtopic is you have to know the importance, you have to know how to draw, you have to know how to form the sentence. Literally is the one that I highlighted over here. If the question asks about subjective question, right, they will ask about chromosomal behavior. Chromosomal behavior, you only need to know that the one that I highlighted over here. Telophase is not a question that they usually ask, so it's okay, you can just know their opposite and prophase. That is good enough already. So the one I highlighted, which is this one, the yellow color one on the right side, that is the important one. So again, our prophase is a preparation phase. Everything started to get ready. Metaphase, they will be at the middle, which is the equatorial plane. Anaphase, they're asking they're being separated. Telophase, they are opposite than prophase. So this is the overall of mitosis. You need to know how to draw. If you don't know how to draw, you will never understand what is the question wanting you to draw. There's a drawing question. There is a forming sentence question when they ask about cell division. Okay, hopefully that this video helps you to understand mitosis. I will continue with meiosis in the next video, hopefully soon. Any question you can comment below, or any parts you don't understand, you can comment below, or any subtopic that you wish to watch, you can also comment below as well. Hope to see you guys soon. See you guys next time. Bye guys. Bye.